So today, I think we'll start with some basics about molecular biology. I know that you are well aware with the structure of DNA, but anyway, I would like to just rewind the things that you have learned in your degree curriculum about the DNA structure, so that if something is missing, that will be complemented in this lecture. And also, while learning molecular biology at PG level, you have to understand the things in a more detailed fashion. There may be a paper or a course in your UG curriculum with the same title, molecular biology. But the depth of the subject that you want to go into, that may be much higher in the case of a PG curriculum. The same DNA replication or the DNA structure, while you are learning, you have to understand a little bit more detailed concepts about that topics. And I will explain how it happens. For example, in case of DNA replication, for example, in UG curriculum, you may be drawing a fork-like structure, just showing a leading and lagging strand and the different molecules that it is in with. That may be the diagram that you are representing. But in PG structure, that may not be sufficient. You have to go into a little more detailed understanding of that particular process and the different orientation of the different strands and the loops that is forming and various other factors that is interacting with the DNA replication. Everything needs to be understood in detail. Similarly, while we are discussing the DNA structure, apart from the things that you already know, you should think why it is so. Why such particular components were used in case of a DNA structure or an RNA structure, so that the overall biology of an organism with a DNA or an RNA and why it is behaving so, everything is well explained in the DNA structure itself, or the components or the chemicals that is used in that particular structure. So you have to understand the things in a more thorough fashion or in a detailed fashion while you are handling a PG course or a PG syllabus. That's what we are. That's what we have to understand initially. Now, in case of a DNA structure, we can see that the DNA is a nucleic acid, which is a, sorry, that slide is shifting. So the DNA is a nucleic acid. And you know that the building blocks of these DNA or RNA, the nucleic acid, they are different nucleotides. And each of these composed of a 5-carbon sugar called deoxyribose in case of DNA, whereas it is a ribose in case of an RNA. And there is a phosphate group or a PO4 group in the DNA molecule, and there is a nitrogenous base. And that nitrogenous base in case of a DNA is a adenine, thymine, cytosine, or guanosine, which may be the thymine is replaced by uracil in case of RNA. That's the only difference that you already know, that is already covered in the UG curriculum. So these are the basic components of a DNA or a nucleic acid, and with slight differences in case of RNA, which is the deoxyribose is replaced with the ribose, and the thymine is replaced with the uracil. That's the only difference in the case of a RNA that we know. Now, in case of a Watson and Crick model of the DNA that we already learned about that model, in that structure, we know that it is a double helical structure. In 1951, James Watson learns about an X-ray crystallography or X-ray diffraction pattern that is studied. It is projected by the DNA. And this study about the X-ray diffraction patterns of the DNA, which was published earlier, and that came into the notice of James Watson in 1951. And the knowledge of the chemical structure of the molecules, that is deoxyribose sugar, the phosphate, and the nitrogenous base. That's the chemical constituents of the DNA that was already existing at the time of this understanding. So James Watson got the DNA X-ray crystallography structure, and there is already existing understanding about the chemical nature of the DNA, which means that it consists of a deoxyribose sugar, phosphate, and nitrogenous base. And there is another rule that is proposed by Erwin Chargaff in his experiments to demonstrate that the ratio of the adenine and thymine is 1:1. And also the guanine to cytosine, that ratio is also 1:1. So that is the Chargaff rule. It's an observation made by Erwin Chargaff and associates. In 1953, James Watson and Francis Crick, they proposed their double helical model of the DNA, or that structure of the DNA, based on these previously existing understandings. And one interesting fact is that this DNA structure that is published in Science, and one interesting fact is that the James Watson and Francis Crick, they didn't perform any wet lab experiments, or they didn't conduct any experiments to prove this hypothesis. They used already existing data or the findings of the previous scientists who worked in that area, and they assembled all these facts and reduced a conclusion. That's what happened. Now, when we come to the detailed chemical structure of the components that is used in case of DNA, we know that there are deoxyribose and ribose sugars that is used in case of a DNA structure. Sorry, DNA or RNA structure. In case of DNA, the sugar that is used is a deoxyribose, and in case of RNA, that is ribose sugar. So the difference is that, and here you can see that maybe okay. This is the OH group that is present in case of DNA or the deoxyribose, and that is present at the 3' position, whereas the 2' position, that is this one I'll show you. So this is the 2' position, 2' position, and the OH is missing, and there is only one H that is present. And in case of the ribose sugar, we have both OH groups that is present at the 2' position and 3' or 2' and 3' positions. So that's the difference between the deoxyribose and ribose. Now, when we come to the bases, nitrogenous bases, we have purines and pyrimidids. Purines are double ringed structures. We have uh, adenosine and guanine. Adenine and guanine, these are the two double ringed structures, nitrogenous bases, which is used in case of nucleic acids. And we have single ringed structures of the nitrogenous bases, that is a cytosine, thymine, and uracil. And you already know that one. Now, why I am presenting this structure in front of you? Because we want to discuss something. Why the 3-OH group, the 2-OH group is missing in case of RNA. I'm placing some questions in front of you. Why the RNA molecule is using an OH group, and that OH group, which is present in the RNA molecule, is missing in case of a deoxyribose that is used in case of DNA. Why this? So what is the biological significance of these removal of the 2-OH group in case of DNA? So I'm placing the question in front of you, like why this OH group of the deoxyribose, the 2-OH group of the deoxyribose is replaced, or it is removed in the DNA, and why the thymine in case of DNA is replaced by uracil in case of an RNA. What is the biological significance of that one? How it affects the overall genetic stability of the DNA and RNA? Now we want to understand that one. That's the main focus of this discussion. And we know that the nucleotide structure, it consists of all these nitrogenous bases, phosphate and hydroxyl group. We already discussed that one. It is only the text portion that is already explained. And we have the phosphodiester bond, which is a bond between adjacent nucleotides. We already know that. And they are formed between the phosphate group of one nucleotide and the 3-OH group of the next nucleotide. That is also understood to us. And the chain of the nucleotides has a 5' to 3' orientation. And it's already a basic fact that we know. So this is that means the sugar phosphate backbone of the nucleotides or the DNA or RNA that is made in the 5' 3' orientation by connecting the phosphate group and the 3'OH group. Now, these are the chemical constituents, as you know. We have a nucleoside, which is when it is attached with the base, and then it becomes the nucleoside, which is without the phosphate, and when it is attached with the phosphate, then it becomes the nucleotide. So the sugar and the base, which constitutes the term nucleoside, and when the phosphate group is attached to that nucleoside, it becomes the nucleotide. This is the chemical constituent that is used in case of a DNA. And the phosphate, sugar and the OH position, that is the 3'. It's quite difficult for me to show you in the case of a mobile device if you are using that one. Because here on the top, on the right side top, we have a nucleotide, and two nucleotides were represented there. And there is a 3'OH group on the nucleotide that is shown on the top side. So that OH will be connected to the phosphate group, which is present in the nucleotide that is represented on the bottom side of the right side bottom. So that bond, that is sugar phosphate, phosphodiester bond, is formed between these two nucleotides to form the phosphodiester backbone of the DNA. So here we have the nucleotide, that is nucleoside plus phosphate, which is represented there. Here we have the Chargaff rule that is already mentioned, because A equal to T and G equal to C, and that percentage is actually it is a 1:1 ratio of these two nucleotides were always present. That is actually discovered by Erwin Chargaff, which is already discussed. And that actually contributes to the foundations of the Watson-Crick model of or that proposal. It is based on these observations of Erwin Chargaff. Now, this is the overall, this table shows the overall ATGC percentages of various organisms. And anyway, I will share the slide with you, so you can go into the details later. So each and every species, if you are taking humans or yeast or some other organism, or wherever you can see the DNA in a species, that species will have a particular proportion of these 4 nucleotides distributed in that genome. So that is, especially we'll say that it is GC content. Because if we state GC content, then the remaining portion will be shared by the AT. So out of the 100 percentage, you have to mention only GC content. So the GC content of a particular organism or a particular species will have some particular range. So each organism will have its own unique ranges of GC content. Okay. Now, the next experiment that we discussed is about the discoveries of the Rosalind Franklin and Maurice Wilkins. And they conducted the X-ray diffraction studies on the DNA structure, and they discovered that it is a helical structure. The DNA is a helical structure. And also they discovered that the molecule has about a diameter of 2 nanometer. And that makes a complete turn within a length of about 3.4 nanometer. So a complete turn of the helical structure or the helical ladder that is at a length of about 3.4 nm, which is a standard dimensions. We already know that. So that observations were actually deduced from the discoveries of Rosalind Franklin, X-ray diffraction study results of Rosalind Franklin and Maurice Wilkins. This is the X-ray crystallography structure of DNA published by Rosalind Franklin et al. And the next one is about the James Watson and Francis Crick proposal of the DNA. That is about that's occurring in 1953. And they deduced that DNA structure, actually using the previously stated evidences, they deduced the structure and they proposed the double helical model of the DNA. The double helical model, actually it consists of 2 sugar phosphate backbones running parallel, and so actually it is anti-parallel. Now, the nitrogenous bases actually it is facing towards the interior of these double strands. So nitrogenous bases were oriented towards the interior portion of these 2 helical ladder-like structure, which means actually it forms the steps of the ladder. And the bases form hydrogen bonds with the complementary bases, or the nitrogenous bases form hydrogen bonds with the complementary bases in the same step of the ladder. And on the opposite sugar phosphate backbone. So this is the purines and pyrimidine pairs that is used. In the case of pyrimidine, you can see that they are single ring structures, and purine pairing, if it occurs, they actually have 4 rings in the structure. But actually, the 20 nm, angstrom or 2 nm size of the DNA, or the width of the DNA, that is actually contributed by a 2-ring structure, that is a purine, and a single ring structure, that is a pyrimidine. So the overall length of the step of the ladder, that is a pair of the nucleotides, that is about 3 ring length, and that length is a 2 nanometer. And if a pyramid pair occurs, the ladder length will be shorter, or the step length will be shorter, which will be a mismatch in the DNA strands. And if you are trying to incorporate or replace a purine with a pyrimidine, and then a purine link will form, and which will have higher length, which will not fit into the normal structure 2nm structure of the ladder. So then also it will produce a bulging or a mismatch, an unstable DNA structure. So in a DNA structure, normal DNA structure, only a pyrimidine pair with 2nm length is only possible. Whereas the shorter one, pyrimidine pair or a purine pair, will not fit into the DNA structure of 2nm width. This is the actual reason for the mismatches that is occurring in case of some errors. If it is occurring in DNA, easily the repair system will identify such mismatches in the length of the DNA structure, which will cause distortions, and that structure will be easily identified, and it is repaired by the DNA repair systems. So this is about the length of the DNA ladder steps or the base pairs, and a purine pyrimidine pair is the only possibility that can be be fixed into the ladder, and not a purine or not a pyrimidine pair, because of this length difference in the actual structure. And actually, if some mismatches occur, certainly that will be identified by the repair systems also. Now, take this example. This is two diagrams that is shown for showing the hydrogen bonding between the nucleotides. And these hydrogen bonds actually it occurs between the bases. And you can see that there are a GC pair, which is shown with 3 hydrogen bonds, and an AT pair with 2 hydrogen bonds, and how it is occurring, that's already demonstrated there. And the main point is that the GC pair is having 3 hydrogen bonds compared to the 2 bonds in the case of a AT pair. So GC pair makes a more stable and stronger bond of 3 hydrogen bonds. So the GC pair will be stronger compared to the AT pairs. So wherever there is a GC rich region in the DNA, that means that region is strongly bound. You will require more temperature to dissociate that one. So the GC rich region is difficult to dissociate, whereas the AT rich regions will be it will be easiest to dissociate or unwind because of the weaker double bonds, or 2 hydrogen bonds between these 2 pairs. So some starting points of some transcription or DNA unwinding, normally we can expect AT pairs where we are expecting a rapid dissociation of DNA strands. There we will have some AT rich regions. Whereas if some regions is not expected to unwind so easily, in that regions where the complementary strands were supposed to bind firmly, and in that regions we will have a higher GC content or GC rich region. So this is about the GC rich regions and AT rich regions, and it is having its own role, biological significance in case of a DNA, especially in DNA unwinding and other things, and even gene expression. So this structure is showing with the thymine and adenine pair, and cytosine and guanine pair, and that's a detailed structure of the previous one. Previously described structure is represented in another manner. Now we come to the phosphodiester bond, which is actually connecting 2 nucleotides. And you know that this is a phosphodiester bond. And it connects the 3' OH group of the deoxyribose sugar and the phosphate group of the next incoming nucleotide. So here, sorry, this structure of the deoxyribose, as a DNA, this phosphodiester backbone of the DNA, where the 2' OH or 2' OH is missing, will be more stable than that of the RNA, the same structure that is produced by the RNA. That's what I am trying to explain. In my previous question that I raised, I asked you why we are using deoxyribose in case of DNA and ribose in case of RNA. Can anybody tell me why we are using deoxyribose in case of DNA and this ribose sugar in case of RNA? The only difference is that we have an additional 2' OH or 2' OH group in case of ribose that is used in RNA. Why this chemical difference? Anybody answer? Hello? Hello? Is the question clear? Can somebody explain or tell me why DNA uses deoxyribose instead of ribose? Hello? Then we'll skip that. I will answer. You can just tell me the answer. Am I audible? Hello? Am I audible?
Yes, sir. You are audible.
Because I thought I'm out of the group because nobody is responding. Eh? So if you can't explain, certainly I will explain. Don't worry. Just answer because since it is a live class, you have to, or I have to, at least I have to feel live, right? Is there any answer for that? No. Okay, fine. Then I will explain. In case of DNA, you know that DNA is supposed to be more stable. It's a stable genetic material that passes the information through generations stably without alteration. Whereas the half-life of DNA is very high. It won't change that easily. It can easily pass from generation to generation without considerable change. Whereas in the case of RNA, what happens? The RNA is degraded very rapidly, maybe within seconds or within minutes, within a minute time. The RNA is produced, immediately utilized, and it is degraded rapidly. And the RNA viruses or the organisms that actually possess this genome, RNA genome, they mutate rapidly, they change. So the possibility of changes in case of RNA or mutation in the case of RNA, and also that what's called the degradation, half-life of the RNA is much higher, right? The half-life is lower or is very short, and the mutation possibility is much higher in case of RNA. So it is very much subjected to rapid change in case of RNA, whereas DNA is much stable. It cannot be easily destroyed or altered, right? So this is the main difference between the RNA and the DNA genomes. Why? Or let me explain why this 2'-OH group is removed from the DNA. In the case of the phosphate, sugar phosphate backbone, the 2'-OH group is actually connected to the phosphate, and that phosphodiester bond, which is actually shown here, the bottom portion, which is connected to the CH2, okay, the CH2O, that bond can be broken. And if another OH is present here at the 2' position, this phosphate will connect to that OH, 2'OH, producing a cyclic nucleotide. So it will break from the chain. That means just a nucleotide chain can be imagined as a chain of people who is holding their hands together for a longer distance. Maybe 10 or 100 people will hold their hands with each other, and you can form a chain. Similarly, the phosphodiester bond is actually both the hands, which is connecting the people, right? So if the O or CH2O is broken, and if you are changing that to the second OH group that is present on the 2'OH group, then it is something like you are holding your own hands. It forms a cyclic one. Actually, these hands were supposed to hold the other one and to form the chain or the backbone. So the phosphate group, that OH will be broken and it will form a phosphodiester bond with the OH, free OH that is present on the 2'OH group, which means wherever and that means a nucleophilic attack. We can say that it is a nucleophilic attack by the 2'OH group will shift the phosphodiester bond to the 2'OH group, resulting in a cyclic structure. And the net result is that the chain will be broken. So you can see these two nucleotides, which is connected by the phosphodiester bond. And if the phosphodiester bond is actually shifts to one of the 2'OH groups of the preceding nucleotide, then the chain will be broken. That means the RNA is destroyed. It is easily degraded. But in case of DNA, that the OH on the 2' position, which is having the ability to do a nucleophilic attack on the phosphodiester bond, is not present in case of DNA. So the DNA is more stable. So you cannot alter the DNA if you are not providing it with another nucleotide or external nucleotide with an OH group to perform a nucleophilic attack. And that is possible. There are some cases like the post-transcriptional modifications where the RNA processing is done. There are some instances something like that, but not in case of DNA. In all the DNA molecules, we have these phosphodiester bond that is formed in between the deoxy sugars, so that the nucleophilic attack with the 2'OH 2' position is not possible in case of DNA, which makes the DNA more stable compared to the RNAs. Is it clear? Hello?
Yes, sir.
Okay.
Yes,
so that's one of the main reasons for the stability of the DNA compared to the RNAs. And if the DNA is incorporated with that 2' OH group, the DNA will be rapidly destroyed, and such diverse genetic information which is carried to the next generation during this evolutionary period will not be possible if the DNA is that much unstable. Okay, fine. Now the next one. So this structure is a similar one, just two nucleotides were shown there. So that's a DNA nucleotide structure and RNA nucleotide structure, which is shown on the bottom. And in the nucleotide structure of the DNA, here we have on the top side, there is no OH group, there is only an H at the 2 position. And on the bottom side, we have the OH group, which is present here, and on the 2' position, which will be able to perform the nucleophilic attack and subsequent degradation of the RNA molecule. Now, This is the phosphodiester bond. So this is the hydrogen bond that is formed between the two uh, DNA strands, and there is a phosphodiester bond that is shown, and the hydrogen bond that is shown, and the purine pyrimidine pairs that is formed, which forms the steps of the ladder in the DNA double helix. All these things were actually well represented in case of this picture. Now, the DNA double helix actually, it's these strands in the DNA double helix. The strands were actually anti-parallel with the sugar phosphate backbone outside the pairs of the bases in the middle. So these double helical structure will have anti-parallel strands. That means one strand is running in 3'5' direction, then the other one will be running in the opposite direction, 5'3' direction. So that means the two strands will be anti-parallel. That is one of the main properties of the DNA double helix. And one is in 5'3' direction, and the other one is on 3'5'. Then two strands that they're wrapped around each other every 30 angstroms. Once with 10 base pairs to create a helical shape of the molecule. So to create a helical shape, the molecule turn around and within a length of 10 base pairs. In a turn of the helix will make a turn within a length of 10 base pairs at about 30 angstroms. And chains are held together by hydrogen bonds between AT and GC pairs. So these are some of the facts about the DNA double helix that is formed. And also here you have another point, that is here is the helical structure, and the ladder or the steps of the ladder which is formed by the purines and pyrimidines. And there is also another concept, that is a major groove and minor groove. So between the steps of the ladder, we have minor groove that is shown here, and the major groove that is represented in this region. Okay, so this is the major groove and this is the minor groove. So this is about the DNA structure. All the turns were represented, and each turn of the DNA molecule, that is so this is the major groove, this is the minor groove, and the one turn will be about 34 angstrom. So this is about the structure of the DNA. The strands are anti-parallel, as you know, and we have a molecular model which is represented here. Then this is the right-handed DNA and the left-handed DNA. The BA-DNA is an example of a right-handed DNA, and the Z-DNA is an example of a left-handed DNA with a zigzag chain. So these are the different forms of the conformational forms or isomers of the DNA, where the B form is a normal conformation, and A form is a conformation that is found at high salt concentrations, and probably is not so biologically relevant. Then D and E forms with 8 and 7 base pairs per turn, and the previously B form actually is having 10 base pairs per turn. There are structures like D and E forms with 8 and 7 base pairs per turn. That model is also there. And DNA segments actually they actually lack guanine in case of D and E forms. Then Z form is there, where we have a left-handed helix with 12 base pairs per turn. It's another model. So P form with that is phosphates towards the inside and bases more towards the outside. So that's another model. So there are various rare forms of conformations of the DNA that is present, and I'm not going into the details. I just want to just brief the various chemical components and the significance of the DNA, particular components in the DNA and RNA structure in this session. So I hope I achieved that one. So the basic concepts of the DNA structure and the different forms of the DNA is already known to you. I'm not going into the details of the different models of the DNA that you can refer some of the previous textbooks. And I didn't answer the second question. That is why thymine is replaced by uracil, or uracil and thymine. That difference is there between the DNA and RNA, right? So in DNA, the thymine is used instead of uracil, and why it is so, that needs to be explained. I didn't explain that one. So here you can see that this is a structure. Here, the last two diagrams, that is thymine and uracil, is mentioned there. So the basic difference is that here there is a CH3 group on thymine, okay, a CH3 group here on thymine, and the remaining is almost same, right? So the difference between thymine and uracil is a CH3 tag on thymine. Okay. Structurally, the difference between thymine and uracil is the presence of a CH3 in thymine, and thymine is used in case of a DNA. Okay. Now, why it is so? Because here we have the cytosine. Okay. The cytosine is having an NH2 group on the top, right? And the deamination process of these NH2 of the cytosine will result in uracil. Okay? The deamination of a cytosine or removal of NH2 by an O will result in production of a uracil. It usually happens because deamination process is a normal process that is occurring in biological systems. And if the NH2 of the thymine or of the cytosine is removed by a deamination process, that results in the production of a uracil. Then what will be the result? For example, in a DNA, we have a cytosine, which is normally paired with the guanine, GC, right? So the deamination will result in removal of that NH2, resulting in the formation of a uracil. Now, if it is left unattended, what happens? It will treat it as uracil or an AT pair equivalent, and then in the subsequent DNA replication step, what happens is that instead of guanine that is expected at that position to pair with the cytosine, the uracil will, or the deaminated cytosine, now it is uracil, it will pair with the A. Okay. So the entire GC pair will be shifting to an AT pair after one or two steps of replication. Is it clear? Hello?
Yes, sir.
So the normally DNA is having cytosine, which is normally paired with guanine, that is GC pair. And if the deamination of the cytosine produces a uracil at that position, in the subsequent replication steps, the uracil will pair with the A. Again, that A will pair with T. So during the next or further replication steps, that AT pair, so the GC pair will be transformed into an AT pair. That is a serious mutation, which is not expected in the system. So DNA, as a stable molecule, it needs to differentiate between these two. That is a deaminated cytosine and a normal thymine which is present in the system, or a uracil which is used in the system. So the DNA actually it uses, instead of the uracil, it uses a thymine, which is actually labeled with another CH3. So if a uracil is identified in a DNA, immediately the repair molecules will come and remove that one. So the pair will not change. The repair system will identify the misplaced deaminated cytosine, and it will be removed by the system, and it will be repaired by the DNA repair systems, so that the JC pair will be maintained within the DNA. There is no possibility of conversion of that GC pair into an AT pair. If DNA uses the uracil, then this deaminated cytosine product uracil and the normal uracil that is used in the DNA cannot be differentiated. So that means immediately it will, all the cytosine, or within time, all the cytosine will be transformed into AT pairs. So that type of modifications were not possible in case of a DNA, because it uses a stable molecule that is a thymine in case of a DNA AT pair. So that's the reason. Okay, now I think both the structural components were explained. I think it's clear. Any doubts? Hello?
No, sir. No, sir.
So let me stop. And I'll make the recordings available in the platform later. And if you have any doubts, you can go through that one. Then let me stop. Okay, bye.